Hey guys, BC here with food for thought. Thank you to all who like, share, comment, and subscribe to my channel. If you're joining for the first time, welcome, and I trust that you find the content useful. Today, we will explore the history, development, testing, and regulations of vaccines. Let's go. Before we begin, I would like to give credit to Dr. Paul Offit from the College of Physicians of Philadelphia for his input into the research and creation of this information. Vaccine development is a long, complex process, often lasting 10 to 15 years, and involving a combination of public and private involvement. The current system for developing, testing, and regulating vaccines developed during the 20th century as the groups involved standardized their procedures and regulations. Government oversight. In the United States, at the end of the 19th century, several vaccines for humans had been developed. They were smallpox, rabies, plague, cholera, and typhoid vaccines. However, no regulation of vaccine production existed. On July 1st, 1902, the U.S. Congress passed an act to regulate the sale of viruses, serums, toxins, and analogous products, later referred to as Biologics Control Act, even though biologics appeared nowhere in the law. This was the first modern federal legislation to control the quality of drugs. This act emerged in part as a response to the 1901 contamination events in St. Louis and Camden involving smallpox vaccine and diphtheria antitoxin. The act created the Hygienic Laboratory of the U.S. Public Health Service to oversee manufacture of biologic drugs. The Hygienic Laboratory eventually became the National Institutes of Health. The Act established the government's right to control the establishments where vaccines were made. The United States Public Service Act of 1944 mandated that the federal government issue license for biologic products, including vaccines. After a poliovirus vaccine accident in 1954, known as the Cutter Incident, the Division of Biologics Standards was formed to oversee vaccine safety and regulation. Later, the DBS was renamed the Bureau of Biologics, and it became part of the Food and Drug Administration. It is now known as the Center for Biologics Evaluation and Research. So, this happened in the US. Elsewhere, in the European Union, the European Medicines Agency supervises regulation of vaccines and other drugs. A committee of the World Health Organization makes recommendations for biologic products used internationally. Many countries have adopted the WHO standards, as always. Stages of vaccine development and testing. In the US, vaccine development and testing follow a standard set of steps. The first stages are exploratory in nature. Regulation and oversight increase as the candidate vaccine makes its way through the process. The first steps: laboratory and animal studies. The exploratory stage involves basic laboratory research and often lasts 2 to 4 years. Federally funded academic and governmental scientists identify natural or synthetic antigens that might help prevent or treat a disease. These antigens could include virus-like particles, weakened viruses or bacteria, weakened bacterial toxins, or other substances derived from the pathogens. In the preclinical studies, tissue culture or cell culture systems and animal testing are used to assess the safety of the candidate vaccine and its immunogenicity or ability to provoke an immune response. Animal subjects may include mice and monkeys. Now, these studies give researchers an idea of the cellular responses they might expect in humans. They might also suggest a safe starting dose for the next phase of research, as well as a safe method of administering the vaccine. Researchers may adapt the candidate vaccine during the preclinical stage to try to make it more effective. They may also do challenge studies with the animals, meaning that they vaccinate the animals and then try to infect them with the target pathogen. Many candidate vaccines never progress beyond this stage because they fail to produce the desired immune response. The preclinical stages often last 1 to 2 years and usually involves researchers in private industries. IND application. A sponsor, usually a private company, submits an application for an investigational new drug, IND, to the U.S. Food and Drug Administration. The sponsor describes the manufacturing and testing processes, summarizes the laboratory reports, and describes the proposed study. An institutional review board, representing an institution where the clinical trial will be conducted, must approve the clinical protocol. The FDA has 30 days to approve the application. Once the IND, investigational new drug application, has been approved, the vaccine is subject to 3 phases of testing. Next steps: clinical studies with human subjects. Phase 1: vaccine trials. This first attempt to assess the candidate vaccine in humans involves a small group of adults, usually between 20 to 80 subjects. If the vaccine is intended for children, researchers will first test adults and then gradually step down the age of the test subjects until they reach their target. Phase 1 trials may be non-blinded, also known as open label, in that the researchers and perhaps the subjects know whether a vaccine or placebo is used. The goals of Phase 1 testing are to assess the safety of the candidate vaccine and to determine the type and extent of immune response that the vaccine provokes. In a small minority of Phase 1 vaccine trials, researchers may use the challenge model, attempting to infect participants with the pathogen after the experimental group has been vaccinated. The participants in these studies are carefully monitored and conditions are carefully controlled. In some cases, an attenuated or modified version of the pathogen is used for the challenge. A promising Phase 1 trial will progress to the next stage. Phase 2 of the vaccine trials. A larger group of several hundred individuals participates in this phase. Some of the individuals may belong to groups at risk of acquiring the disease. These trials are randomized and well controlled and include a placebo group. The goals of Phase 2 testing are to study the candidate vaccine's safety, its immunogenicity, proposed doses, schedule of immunizations, and method of delivery. Phase 3 vaccine trials. Now, successful Phase 2 candidate vaccines move on to larger trials involving thousands to tens of thousands of people. These Phase 3 tests are randomized and double-blind and involve the experimental vaccine being tested against a placebo. The placebo may be a saline solution, a vaccine for another disease, or some other substance. One Phase 3 goal is to assess vaccine safety in a large group of people. Certain rare side effects might not surface in the smaller groups of subjects tested in earlier phases. For example, suppose that an adverse event related to a candidate vaccine might occur in 1 of every 10,000 people. To detect a significant difference for a low-frequency event, the trial would have to include 60,000 subjects, half of them in the control or no vaccine group. Vaccine efficacy is tested as well. These factors might include: 1. Does the candidate vaccine prevent disease? 2. Does it prevent infection with the pathogen? And 3. Does it lead to production of antibodies or other types of immune responses related to the pathogen? Next steps: approval and licensure. After a successful Phase 3 trial, the vaccine developer will submit a biologics license application to the FDA. Then, the FDA will inspect the factory where the vaccine will be made and approve the labeling of the vaccine. After licensure, the FDA will continue to monitor the production of the vaccine, including inspecting facilities and reviewing the manufacturer's tests of lots of vaccine for potency, safety, and purity. The FDA has the right to conduct its own testing of manufacturers' vaccines. Post-licensure monitoring of vaccine. A variety of systems monitor vaccines after they have been approved. They include Phase 4 trials, the Vaccine Adverse Event Reporting System, and the Vaccine Safety Data Link. Phase 4 trials. Phase 4 trials are optional studies that drug companies may conduct after a vaccine is released. The manufacturer may continue to test the vaccine for safety, efficacy, and other potential uses. The CDC and FDA established the Vaccine Adverse Event Reporting System, VAERS, in 1990. The goal of VAERS, according to the CDC, is to detect possible signals of adverse events associated with vaccines. A signal in this case is evidence of a possible adverse event that emerges in the data collected. About 30,000 events are reported each year to VAERS. Between 10% and 15% of these reports describe serious medical events that result in hospitalization, life-threatening illnesses, disability, or death. VAERS is a voluntary reporting system. Anyone, such as a parent, a healthcare provider, or a friend of a patient who suspects an association between a vaccination and an adverse event may report that event and information about it to VAERS. The CDC then investigates the event and tries to find out whether the adverse event was in fact caused by the vaccination. The CDC states that they monitor VAERS data to detect new, unusual, or rare vaccine adverse events, to monitor increases in known adverse events, to identify potential patient risk factors for particular types of adverse events, to identify vaccine lots with increased numbers of types of reported adverse events, to assess the safety of newly licensed vaccines. Not all adverse events reported to VAERS are in fact caused by a vaccination. The two occurrences may be related in time only, and it is probable that not all adverse events resulting from vaccination are reported to VAERS. The CDC states that many adverse events, such as swelling at the injection site, are unreported or underreported. Evidence exists that the Chinese employed smallpox inoculation as early as 1000 CE. It was practiced in Africa and Turkey as well before it spread to Europe and the Americas. Edward Jenner's innovations began with the successful 1796 use of cowpox material to create immunity to smallpox, quickly made the practice widespread. His method underwent medical and technological changes over the next 200 years and eventually resulted in the eradication of smallpox. Where did smallpox come from in the first case? Louis Pasteur's 1885 rabies vaccine was the next to make an impact on human disease. And then, at the dawn of bacteriology, developments rapidly followed. Antitoxins and vaccines against diphtheria, tetanus, anthrax, cholera, plague, typhoid, tuberculosis, and more were developed through the 1930s. And then you'll have to tell us, where did all of these viruses come from? Nature? The middle of the 20th century was an active time for vaccine research and development. Methods for growing viruses in the laboratory led to rapid discoveries and innovations, including the creation of vaccines for polio. Researchers targeted other common childhood diseases, such as measles, mumps, and rubella, and vaccines for these diseases reduced the disease burden greatly. I wonder where all of those diseases went to, because I'm not hearing of them now. Is it because of the vaccines? The vaccines have eradicated them? Think, don't just believe. Innovative techniques now drive vaccine research with recombinant DNA. With recombinant DNA technology and new delivery techniques, leading scientists in new directions. Disease targets have expanded, and some vaccine research is beginning to focus on non-infectious conditions such as addiction and allergies. Why are people addicted? Why are people addicted, and what are they addicted to? Oh, I see. They are addicted to things that were specially designed for addiction. And so are those that cause allergies. It's not by any mistake. Okay? More than the science behind vaccines, these timelines cover cultural aspects of vaccination as well. From the early harassment of smallpox to the establishment of vaccination mandates, to the effect of war and social unrest on vaccine preventable diseases. All of these creatures who have led hands in the pioneering of vaccines, they have a lot of questions to answer to the small man, to those who are not scientifically oriented. It's funny how in science they have so many big words that you catch your hell to even pronounce them. If these words really do have a meaning, and maybe it's just a deterrence so that people like myself and others wouldn't even dare venture into those boundaries. Alright? But there are many questions. We have many questions, but no one is gonna answer us. Let's look at the recombinant DNA. What really that is. Simply put, our DNA. They are DNA molecules formed from laboratory methods of genetic recombination, right? Such as molecular cloning. Have you ever heard that word before? Cloning? That bring together genetic material from multiple sources. So take from meat, take from the dog, take from the plant, and merge them all together, right? Creating sequences that would not otherwise be found in the genome, right? So what's the reason for this? What's the reason for merging or splicing DNA from different sources? Is it to make mankind better? Or maybe it's for vaccination, so that we can conquer those contaminating, those pandemics, right? Recombinant DNA is the general name for a piece of DNA that has been created by combining at least two fragments from two different sources. Recombinant DNA is possible because DNA molecules from all organisms share the same chemical structure and differ only in the nucleotide sequence within the identical overall structure. Recombinant DNA molecules are sometimes called chimeric DNA or chimeric DNA because they can be made of material from two different species, like the mythical chimera. Our DNA technology uses palindromic sequences and leads to the production of sticky and blunt ends. The DNA sequences used in the construction of recombinant DNA molecules can originate from any species. For example, plant DNA may be joined to bacterial DNA, or human DNA may be joined with fungal DNA. In addition, DNA sequences that do not occur anywhere in nature may be created. Wow! Genius, isn't it? To be able to create something that doesn't exist in nature. And this is only done through a chemical synthesis of DNA and incorporated into recombinant molecules. Using recombinant DNA technology and synthetic DNA, literally any DNA sequence may be created and introduced into any of a very wide range of living organisms. So I understand that now. So create a new structure that doesn't exist and then put it into what is natural for the purpose of modifying, genetic modification. Proteins that can result from the expression of recombinant DNA within living cells are termed recombinant proteins. When recombinant DNA encoding a protein is introduced into a host organism, the recombinant protein is not necessarily produced. I'll read that again. When recombinant DNA encoding a protein is introduced into a host organism, the recombinant protein is not necessarily produced. Expression of foreign proteins requires the use of specialized expression vectors and often necessitates significant restructuring by foreign encoding sequences. You heard that? It necessitates significant restructuring. Now, recombinant DNA differs from genetic recombination in that the former results from artificial methods in the test tubes, while the latter is a formal biological process that results in the remixing of existing DNA sequences in essentially all organisms. You get that? Recombinant DNA differs from genetic recombination. The former is artificial, the latter is natural. Okay? What have you learned? Question: how long does the vaccination developmental process last? How long does the vaccine development process often last? Who is responsible for issuing license for vaccines in the US? Who is responsible for issuing license of vaccines in the US? And thirdly, true or false? Vaccines are tested for safety in animals before they are tested in humans. Again, vaccines are tested for safety in animals before they are tested in humans. True or false? Don't think, just believe. Just believe. Vaccines were not developed for animals nor human safety. I repeat, vaccines were not developed for animals nor human safety. If they were, the testing subjects would have been some other entity, maybe plants. But the fact that they are testing it on animals and humans, they're not for our safety. Don't think, just believe. Vaccines are a form of biological weapon used in the control of population growth. I repeat, vaccines are a form of biological weapon used in the control of population growth. Think, don't just believe. Many are walking around asking the question, are you going to take the vaccine? What good does that bring? Whether my answer is yes or no, how will that help you? Worry not yourself about who's going to take the vaccine. You worry about you and what decision you're going to make and how it's going to affect you and your future generation. Your DNA has already been modified and it continues to be modified, right? And if you continue to allow it to be modified, that will be your choice, not no one else's. So stop asking people if they're going to take the vaccine or not. That's not your business, okay? You make a difference in you. Do not allow someone else's choice to affect the way you feel, right? Stop believing and start thinking. There's way too much information out there, way too much. The evidences are immeasurable. So you don't need to be investigating people, asking them if they're going to be taking vaccine or not. Do your research and you find out why is it that vaccines have been stopped testing since the 70s. Why have they stopped testing vaccines since the 70s? You need to find out why, and then you will know that since then no vaccine has been tested, none. So you be a subject, you be a human subject, and allow yourself to be a guinea pig and let them test it on you. The data is needed, so at least the rest of us will know the efficacy of it. Okay? I hope this information proved you well, served you well. Continue to be focused, continue to be safe, and think, don't just believe.